Ganglia Part 1 Tail Pallidum. Basal ganglia is a group of subcortical nuclei of different origin. Basal ganglia is located at the base of the forebrain. Basal ganglia are strongly interconnected with cerebral cortex, thalamus, and brain stem, as well as several other brain regions. Basal ganglia are associated with variety of functions, which includes control of voluntary motor movements, procedural learning, routine behavior or habits such as teeth grinding, eye movements, cognition, and emotions. Components of the basal ganglia: the striatum, both dorsal striatum, which includes caudate nucleus and putamen, and ventral striatum, which includes nucleus accumbens and olfactory tubercle. Other parts are globus pallidus, ventral pallidum, substantia nigra, and subthalamic nucleus. Striatum receives input from many brain areas beyond the basal ganglia, but only sends output to other components of the basal ganglia. The pallidum, it receives input from the striatum and sends inhibitory output to a number of motor related areas. The substantia nigra is the source of the striatal input of the neurotransmitter dopamine, which play an important role in basal ganglia function. The subthalamic nucleus receives input mainly from the striatum and cerebral cortex and projects to the globus pallidus. It helps to decide which of the several possible behaviors to execute at any given time. The basal ganglia's primary function is likely to control and regulate activities of the motor and premotor cortical areas so that voluntary movements can be performed smoothly. The basal ganglia exert an inhibitory influence on a number of motor systems, and that a release of this inhibition permits a motor system to become active. The behavior switching that takes place within the basal ganglia is influenced by signal from many parts of the brain, including the prefrontal cortex, which plays a key role in executive functions. The importance of these subcortical nuclei for normal brain function and behavior is emphasized by the numerous and diverse neurological conditions associated with basal ganglia dysfunction, which include disorders of behavior control such as Tourette syndrome, hemiplegismus, and obsessive compulsion disorder, dystonia, addiction, and movement disorders, the most notable of which are Parkinson's disease, which involves degeneration of the dopamine producing cells in the substantia nigra, pars compacta, and Huntington's disease, which primarily involves damage to the striatum. The basal ganglia have a limbic sector whose components are assigned distinct names: the nucleus accumbens, ventral pallidum, and ventral tegmental area (VTA). There is considerable evidence that this limbic part plays a central role in reward learning, particularly a pathway, mesolimbic pathway, from the VTA to the nucleus accumbens that uses the neurotransmitter dopamine. A number of highly addictive drugs, including cocaine, amphetamine, and nicotine, are thought to work by increasing the efficacy of this dopamine signal. There is also evidence implicating overactivity of the VTA, dopaminergic projection, in schizophrenia. Here we can see the chart which tells about different parts of the brain and their origin. The basal ganglia form a fundamental component of the cerebrum. In contrast to the cortical layer that lines the surface of the forebrain, the basal ganglia are a collection of distinct masses of gray matter lying deep in the brain, not far from the junction of the thalamus. They lie to the side of and surround the thalamus. Like most parts of the brain, the basal ganglia consist of left and right sides that are virtual mirror images of each other. The subthalamic nucleus and substantia nigra lie further back posteriorly in the brain than the striatum and pallidum. Striatum and pallidum are larger, while the subthalamic nucleus and substantia nigra are smaller. Coronal section of the human brain, you can see here the striatum and substantia nigra this side and subthalamic nucleus here. Here also we can see the nucleus accumbens caudate. Caudate, it has three parts: tail, body and head. This is putamen, globus pallidus and thalamus. Striatum. The striatum is the largest structure of the basal ganglia. The striatum, also the neostriatum and the striate nucleus, is one of the nuclei in the subcortical basal ganglia of the forebrain. The striatum is a critical component of the motor and reward systems, receives glutamatergic and dopaminergic inputs from different sources, and serves as the primary input to the rest of the basal ganglia nuclei. Functionally, the striatum coordinates multiple aspects of cognition, including motor and action planning, decision making, motivation, reinforcement, and reward perception. The striatum, as seen on MRI, the striatum in red includes the caudate nucleus, top, the putamen, right, and when including the term corpus striatum, the globus pallidus, lower left. Structurally, when the globus pallidus, pale globe, is included with the striatum, they form the corpus striatum structure. The lentiform nucleus refers to the putamen of the dorsal striatum and the globus pallidus. Based on the function and connection, the striatum is divided into ventral and dorsal subdivisions. The ventral striatum consists nucleus accumbens and the olfactory tubercle. The dorsal striatum consists of the caudate nucleus and putamen. A white matter nerve tract in the dorsal part separates the caudate nucleus and the putamen. The striatum is termed for stripped appearance of white and gray matter. Nucleus accumbens. Nucleus accumbens is made up of nucleus accumbens core and nucleus accumbens cell. These both differs by neuron populations. The olfactory tubercle. Olfactory tubercle receives input from the olfactory tubercle, but it doesn't demonstrate processing of smell. In the non-primates, islands of Calaza are found here. Ventral striatum is associated with the limbic system, and it is a major part of circuitry for decision making and reward related behavior. Dorsal striatum. Dorsal striatum is composed of caudate nucleus and putamen. Staining can differentiate the dorsal striatum into components like striosomes and surrounding matrix. Striosomes, striatal patches. This is one of the two chemical compartments of the striatum. Another one is matrix. These can be visualized by staining for immunohistochemical markers. Immunohistochemical markers like acetylcholinesterase, enkephalin, substance P,